speaker is Dr. Timothy Hain, and I suggested to Tim I was going to make some stuff up. So Tim studied with Luciano Pavarotti in the Opera House of Milan, and actually sang Nessun Dorma with Luciano in a really amazing duet in the 70s. He studied with Emeril Lagasse, and actually taught Emeril how to cook Creole and Cajun cuisine. When he was sort of formatively working in Louisiana. I think that's all I can think of.
That sounds great. Yeah, that's great. Yeah.
Actually, Tim is a very accomplished fish biologist and behavioral ecologist. He's an assistant professor at the University of Western Ontario. He teaches a suite of courses in ecology and environmental science. He did his PhD actually at Western and was working with longtime researcher of QUBS, Brian Neff. And it says here, Tim is in his 12th field season at QUBS, which is amazing. And he is interested in social and mating behavior of bluegill sunfish. He's worked on both guppies in Trinidad, if I'm not mistaken, and also sunfish here. And it has worked a lot on kin recognition, has done some really cool work on kin recognition fish and made some really important discoveries in that domain. And has published a number of well-regarded papers if you go to his Google profile. And also I found, because I went to the fieldwork blog, you published a bit of a memoir recollection of your time in Trinidad.
I did, yes.
Very much liked. And so I encourage you to go look it up. I saw there's a Kindle edition on Amazon at least, and it's called Fieldwork Stories from Trinidad. But today, Tim is going to talk a little bit about musings on 40 years of bluegill research that's been done. A lot of it has been done here. So thank you very much.
Great. Hey. Great. Thanks very much, Steve. I'll add that Emeril changed my recipe. I had originally had the Creole with bluegill in it, and he adjusted it to make it more shellfish based. Yeah, and my book is available on Google or on Amazon. I've sold 2 copies. You can be the 3rd buyer of my book. Today I'm going to be talking about bluegill research. And one thing that Steve didn't mention is that I grew up about an hour away from here on the St. Lawrence River in Brockville. And so I grew up fishing for a lot of the same fish that are in our lake here. And when I was a kid, actually I'll take this off, sorry. When I was a kid catching fish, I imagined being caught and seeing the air being the most exciting part of their day. I couldn't fathom that they would have a more interesting life under the sea, or in freshwater, not in the sea. And then when I started to study them, I realized, wow, these fish actually have some really interesting behaviors and really interesting lives. And so I had to adjust my understanding of how fish live their lives. And in the same way, there's been some research over the last 40 years that has made us re-examine a lot of things that we thought we knew about bluegill. And that's one thing that's really exciting to me about these long-term studies, is that we see how certain species can adjust, and we can re-examine some assumptions we've made about the species. And one message I'd like to, or 2 take-home messages I'd like to give to you about bluegill are that they are a very good species at reacting to environmental stressors. And perhaps a more general message, I'd like you to think about taking some received wisdom about your study species and not treating it dogmatically as if it's something that needs to be held firm. You can adjust some of your assumptions. So that's something I'll be talking about today. This is a picture of a male bluegill sunfish on its nest. They're a very pretty fish. They grow to be about 21 centimeters long in this lake. This is its range in Ontario. And this is its range in the continental United States. So of course, QUBS is sort of in this area here where it is part of the range, but we're sort of near the northern part of the range for the bluegill here. Bluegill here in this lake are sympatric with pumpkinseed sunfish. So pumpkinseed are also found in this lake, Pumpkinseed typically extend their range north of here, and where we are is near the southern end of their range. Pumpkinseed and bluegill do hybridize, and there's been a lot of work done by a former QUBS researcher, John Epifanio, to look at the hybridization of bluegill and pumpkinseed. He's found that the pumpkinseed can produce sperm, but they're really bad at using it. And so typically in a nest of hybrid sunfish, you'll see most of those offspring were fertilized by bluegill sunfish who build nests very close to those hybrids. Now, over 40 years, there have been a lot of researchers collecting data, and I've been up here for 12 years, but those aren't 12 continuous years. My first year up here was 2004. So there were a lot of years when I wasn't here where some people picked up the slack. The first researcher who I'm aware of that did bluegill research at QUBS was Alan Keast. And Alan has a cabin named after him over that direction. He did a lot of work on both birds and sunfish and other fishes. He was not part of the pedigree or the family tree that's shown for the rest of this slide. Who I think of as my academic grandfather was Mark Gross. Mark did his work out of the University of Toronto, but did all of his field work here. And he published a really important paper on bluegill life histories in 1982, hence the 40 years of research here at Western. He had a number of students, including my supervisor, Brian Neff, who is at the University of Western Ontario, and he collaborated with Rosemary Knapp. She first came up here in 2004, and she was really instrumental in my development as a researcher, and also Emma Churchman, who was up here in 2018 to 2021, working on how hormones affect the reproductive behavior of bluegill. I'm also going to be talking about some of Scott Colborn's work. He's now a postdoc at the University of Michigan, and I'll talk a little bit about stuff that I did, but I'm not going to be talking too much about my own published research. This is meant to be more of a fun musings kind of talk. Of course, there are many others who have worked on this. Dave Phillip, who will be speaking to you next week if things go well, also has done a lot of really important work on bluegill, and I've already mentioned John Epifanio's name. Now, that first name on that list, Alan Keast, had invested his time partly in understanding the diet of bluegill and pumpkinseed sunfish. He was interested in how these two species that look very similar and occupy this lake can coexist. Do they have overlapping niches? And in what way do they differ? So to look at that, he examined the diets of pumpkinseed and bluegill sunfish for many years of their life. So each of these Roman numerals represents the age of the fish in years. We have age 0 fish up to age 6+ fish. And there are a whole bunch of invertebrate orders listed here. The big thing that I'd like you to take from that is that they're both generalists. They eat a lot of different things, but there are a few areas where they differ. Most notably, they differ in their ingestion of gastropods, so the snails. Snails, of course, have very hard shells, so that makes it hard for a lot of fish to crush them. Pumpkinseed sunfish have mouths that are a little more downward facing, and their mouths are a little bit bigger, their jaws are a little bit stronger, so they're really good at crushing snail shells. Bluegill, on the other hand, do not have those jaws. They don't really eat gastropods or other hard-shelled organisms. But in general, they eat a lot of zooplankton, particularly the larvae. The age 0 individuals eat a lot of Cladocera, as well as other floating plankton. All plankton floats. Now, this is important. I'd like you to remember that pumpkin seeds like hard-shelled things. Bluegills don't really go for it. Now, typically in the lake, we see bluegill dig nests and form colonies like this. So anyone who puts on snorkeling gear will have an opportunity to see these fish. It's going to run. My video will run or not here. Okay, there it's going to run. So this is a snorkeling image I took of a bluegill colony. They have all these nests that are these hollowed out circles. And these fish on the nests are male bluegill sunfish. They dig these nests in colonies, often rim to rim, and they court females and spawn with the females. And then they're left to care for the young. I'll point out this individual. Maybe you saw it. Maybe I'll just go back for a moment so you can see it again. This one individual, if I go way up here. Okay, we're not going to let me, it's not going to let me fast forward, but it's significant. So let's watch that one individual again. We'll see that there's some circling the nests. There are some that are, waiting for females. Some try to attract females by showing off how good a parent they will be. Part of that is defending the nest by circling. This guy is sweeping out his nest. That's one way that the fish aerate their eggs and prevent waste products from building up near the eggs, and that can reduce the survivorship of the young. So in a bluegill colony, there's a lot of activity, and it can lead to just a naturalist having a lot of curiosity about the behavior of these sunfish. So let's move on. And it's because of this curiosity that Mark Gross looked at the life histories of bluegill. So in his 1982 paper, he had looked at different sizes and different ages of bluegill sunfish. And one thing he noticed is that, sure, there are those males that dig the nests and then they court the females, spawn with them, provide care for the young. But then there are a lot of males who seem to be getting into the nests, but don't seem to be sticking around to care for the young. So he looked at the gonadal development of the young. He looked at the sizes at different ages of the two different types of males. There are males who he called parentals, and males who he called cuckolders. The males who he called parentals were immature. Their gonad development was very limited until they were 7 years old. And then after 7 years, they were active, they were mature, they dug the nest, they cared for the young. The cuckolder males were not involved in parental care. They would mature as early as 2 years of age and adopt a sneaking tactic. So if they saw a male and a female in a nest spawning, they would dash into the nest, release their sperm, and take off, leaving the parental male to care for the young. As these males get older and bigger, they're not as good at hiding on the periphery of the nest. So instead they adopt what's called a female mimic tactic. They look a little more like a female. They're a bit smaller still, so to our eyes, they're often easy to tell apart, but they behave a lot like a female bluegill, and their hormone profile is very similar as well. So we do think that they smell a lot like a female bluegill as well. As a female mimic, they'll wait until when they see a male and a female in a nest together, and when they do, they'll sort of wedge themselves in between, and as the female's dipping on her side to release eggs, the mimic will release its sperm, and again, leave the care to the parental male. So I'll show you the data on how successful those different reproductive tactics and strategies are a little bit later. Typically, the parental male wins where they have more than half of the eggs in their nest, but in any single dip by female, if a cuckolder intrudes, the cuckolder does fertilize more of the eggs. Parental care is very costly, so you can see why, or you might be able to imagine why these cuckolder males have an advantage. They try to avoid some of those costs of parental care by leaving the care to these males. And some studies have shown that these nest-tending parental males lose up to 10% of their body weight during the caregiving period. And so this video will show you some of the care that these parental males do as they're caring for these eggs and then the larvae. So they're circling the eggs a lot. If they see an intruder, they'll dash towards it. They may flare their opercular flaps or their dorsal fin. They may try to attack the intruder. They do fan it like this male is doing. Sonia's kids describe this as looking like they're shaking their booty, and I do agree that that's a pretty good description of it. So this is a picture of everything that's going on with the bluegill. We have the male, the parental male here on his nest. He has a female. We don't really see her in profile because she's dipping on her side. She's about to release some eggs. Beneath this female, we have a sneaker male. He's gotten himself in a nice position where he's going to be really close to where the eggs are. He can release his sperm, and he usually gets about 80% of the fertilizations in a single dip if he's entered the nest. On the side, we have a female mimic. She's not in a good position to fertilize the eggs, but in another dip, maybe she would be. And she fertilizes about 2/3 of the eggs when in competition with a parental male. So this is a short video that shows a spawning day for bluegill sunfish. We see this parental male. He's built his nest. There's a number on it. We use numbers on our tags that are on our nest that help us identify which male we're looking at. Because males are so philopatric, we don't need to tag the fish themselves. They keep returning to their nest. So we know that this individual, 56, is going to be there for as long as the care period happens. So let's watch this video. So there is a female. There is a much smaller female mimic. You can see him get in between the two, and this male is trying to chase away some of the sneakers, seemingly unaware that there is this one female mimic in the nest that he seems fairly comfortable having in his nest. He doesn't mind having what he thinks is a second female in his nest because, wow, that's more eggs. That's a good thing for me. He's devoting all of his extra time chasing to these sneakers on the periphery. It's not unusual for a male to have more than one female in his nest at a time. And in this picture, we can see this male has 3 females and a mimic in his nest at a time. So he's doing very well. He's getting a lot of eggs in this one event. And so for him to try to discriminate between the female mimics and the females means that he would lose out on a lot of opportunity to fertilize eggs if he disrupts the whole thing. There are plenty of other males there who would be happy to have those females in their nest. So that's some background on the life history of bluegill. Oops. Mark Gross had done this research to look at how bluegill life histories develop. He had taken this picture in the early 1980s when camera technology was not as good as it is today. But you can see that there's a bunch of sandy nests there built rim to rim. There is a female in this nest spawning with the male. There's not a lot of vegetation in this lake, but he had started studying this population about 40 years ago. And some of his earliest papers on bluegill talked about huge colony sizes. These are some screenshots that I took of a couple of papers, one published in 1979, so even before this period, and then his key paper in 1982. So 1977, he talked about 2 colonies having about 75 males each. And here he wrote that some colonies would have 5 to 150 colonies at a time. And those numbers boggle my mind. I have not seen a colony that big in a long time. Nowadays, a colony of 30 nests is most common. So this suggests that maybe the population size of bluegill is not as high as it once was. We haven't done any rigorous population estimates for this lake, but it does suggest that the population size is going down. And that could be of concern to some fisheries biologists or conservation biologists. One thing that is interesting that sort of counterbalances that trend is that the average size of an adult male is going up. So in 1982, Mark Gross had reported that the average size of a parental male was about 17 centimeters. I've started collecting data over 15 years ago. Here's some data that I collected from bluegill 2005, 2008, 2020. Hasn't really been much of a change in the average male size in the period that I've been here, but it's grown by maybe 15% since 1982. And the fish are significantly bigger than they were back then. So smaller population size, but each individual fish is getting bigger. I'll let you think about what has changed in the lake since the 1980s. Do you want to guess? Absolutely, yeah. Thank you. You've sort of read my mind and anticipated what this next slide is. So there are zebra mussels in the lake now. One thing that zebra mussels eat is zooplankton. They eat a lot of the stuff that is filtered out of the water. And so this means that we have an important competitor with baby bluegill in the first year of life. Something else that has changed since the early 1980s is that fish have started to eat zebra mussels. So I can tell you that when I came here in 2004, you could not walk on the rocks in Lake Opinicon without slicing up your feet. Nowadays, if you step carefully, you can get away without wearing shoes in the water. You still have to be a little bit careful because there are still zebra mussels here, but the population size of zebra mussels has gone down even since then. And one reason why, perhaps, is that some of the local fish have learned to eat zebra mussels. As I mentioned earlier, pumpkinseed, who are in this lake, have jaws that are really good at crushing snails. Scott Colborn, who I introduced you to earlier, showed us that pumpkin seeds do eat zebra mussels. So he looked at diet of pumpkin seeds both by looking inside their gut and also by doing an isotope analysis. And he found that in Lake Opinicon, about 3/4 of the diet of pumpkin seed came from zebra mussels. And that's pretty much the same story in other lakes in the Rideau system. So boats have brought in zebra mussels, but the local fishes have adjusted their diet to drop the risk of zebra mussels, competing with some of our young bluegill. So the message I see from this is that we do have a lot more competition for food early in life for bluegill. So this picture shows us a whole bunch of baby bluegill. They're about 10 days old. They haven't left their nest yet. Tiny little things. And of course, zebra mussels are competing with those larvae for food. But zebra mussels may also be a food source for adult bluegill. So there seems to be competition for food with baby bluegill. But once they're through this period of competition, there's less interest-specific competition among older bluegill, and so they have an ability to grow bigger, and they may even have the ability to consume zebra mussels. There's been some anecdotal evidence to suggest that bluegill eat zebra mussels as well, but not studied as rigorously as Scott did with the pumpkinseed. When we look at the feces, we do see shells of zebra mussels coming out, and a couple of fishers have reported zebra mussels in the gut of bluegill. So that's one way that bluegill have been able to adjust to an environmental stressor. They've been able to perhaps change their diet to take advantage of a potential food source. Another thing that's happening in this lake is that it is getting warmer. So again, this is something that's not studied rigorously, but it's something that I am curious about, and I wish we had started to study it rigorously back when we had some baseline data. So typically what happens is that fish spawn, then after 10 days, the eggs hatch. So these eggs, as you can sort of make out, look kind of like cooked quinoa. And I know that some students have used quinoa as a visual rather than try to photograph eggs to put in their presentations. And then after 10 days, the larvae disperse from the nest. So this is a picture of baby bluegill, 10 days old. They look like maybe grains of rice with eyes, and they're hopping around in the nest. So we have studied bluegill for 40 years, and we worked with this received wisdom. Emma Churchman, whose name I had shown you earlier, designed her PhD project with this knowledge. She thought I would have 2 days to work with bluegill before the eggs hatch, and then I would have up to 7 days to work with the bluegill before they leave the nest. But in recent years, this has not been the case. In fact, by June 20th, we see an acceleration in development. So this was discovered because Emma wanted to collect data on day 1 and day 2 before the eggs hatched, and then she wanted to collect data on day 4 and day 5, the day after eggs hatched. What she found was that when we went to the colony on day 2, the eggs had already hatched. We lost a day to be able to study them. So these eggs are hatching a day early, and the larvae are dispersing up to 3 days earlier than they had historically. So this does suggest that warming in the lake has led to faster development of these bluegill. How does that affect the population? We don't know yet. So one other thing I wanted to share with you is some of these data on how successful bluegill are at fertilizing the eggs. These parental males are in competition with cuckolders, and most of the time the parental males have no problem being able to fertilize eggs without being in competition with cuckolders. But there have been several studies that have looked at paternity of parentals. One of the first ones was done by my supervisor, Brian Neff. For his thesis in 1996, I think, he took a huge net and blocked off Keast Beach or Sandy Bay. No fish could come in and come out, or come in or out. And so he was able to track every single fish that had set up a nest in this colony. And he drew a map that looks like this. Each of these circles represents a nest in a colony. These dark spots represent a rock. This is a log, this horizontal, well, I know, rectangular thing for people watching at home. And each of the circles may have a number in it if the males stayed there until the eggs hatched and the larvae left the nest. There are some nests that were abandoned by males, perhaps because they didn't get enough eggs for it to be worthwhile, perhaps because they weren't in good enough condition to make it through the 10 days. So you can see that there are a few males who were very successful. They had 100% paternity in the nests, but there are some males that did not do very well. The lowest one had 26% paternity. I'm having, oh, it's down here, 32, 38, some very low numbers, and on average, 79% paternity by parentals. There have been 2 other studies I'm aware of since then, one by Dave Phillip, that have consistently shown between 79 and 83% paternity by the parental male. But there were a lot of nests that were abandoned before the end of the period, the caregiving period. And what happens to the larvae in those nests? Well, often those nests were abandoned because there weren't enough eggs for it to be worthwhile for the male to stick around. But it's been sort of received wisdom that any larvae that were in the nest when the male left would get eaten. There's no chance of survival for those eggs or larvae if their father left. So it was assumed that any larvae that were left behind were quickly eaten by snails or bullhead catfish, or most often other sunfish. And when I work with the bluegill on these nests, I quickly put down nest covers made with chicken wire and duct tape to prevent fish from getting in. But even the smallest sunfish can very easily get under that chicken wire. So I can see how very quickly some of these larvae would disappear if not for that male to protect the nest. But some research over the last few years has uncovered or revealed that there is another way. There's another way for these eggs to survive. So Emma Churchman, who was here for a few years, monitored eggs up to 5 days after spawning. So we had these numbers on the nest to be able to track these nests, these males. Oftentimes we'd come back after a day or 2 and males had left. So the fish was gone. We didn't see any larvae or eggs in there. But sometimes we found that there were larvae in a nest, even though there didn't seem to be that one male still there. So this video shows us some alloparental care by one sunfish. You can see that this guy is actually protecting 2 different nests. Here he is on one nest. He sort of inspected it, spending some time here. And then he's going to work his way back to the other nest. So here it's possible for males to protect more than one nest. We saw this for the first time in 2018, and we observed it 2 more times in 2021. So this is not a common behavior, but it does occur where there's this care by another male to protect the eggs. Why does that behavior occur? I'm open to ideas, but some possibilities are maybe this male was related to this male, and so there's some kin selective benefit to protecting both nests. As someone who did his PhD on kin recognition, that's an idea that's really exciting to me. It's possible that they have elevated levels of prolactin, which is a hormone associated with parental care. It's also possible that he is directly benefiting by perhaps pecking away at the food in this nest when he needs a food source. There are other possibilities, and it's kind of exciting to think about. So some of the takeaway messages from this talk are that bluegill seem to be responsive to their environment. The zebra mussels have led to changes in the body size, but also to the colony size, maybe even the population size of bluegill sunfish. We've seen that warmer water, perhaps because of climate change, has led to changes in development times. That's a pretty common idea with ectothermic animals. And another lesson that I want to really share with you is that we should try to avoid describing a species ecology dogmatically, where we take received wisdom and just expect it to be true forever. Something we've seen is that just by haphazard observations, some of our assumptions were wrong. So we did see alloparental care once for the first time in 2018, twice in 2021. I can't remember if I said that date correctly or not previously, but there's the correct date. So that's it for today. I'd like to acknowledge everyone at QUBS for hosting us for many years, 40 years, I guess. Emma is shown here. Peter Baker is now doing his master's at Western. He was up in 2020 when the aquarium facility first came down. I don't really know what we're doing here, but it's not helping things. So thanks for your attention. I'll take any questions.